Measles and rabies. Yeah? Good? That's what we get to talk about today? But before that, what do we have to do? Exactly. So, clicker question 14.1. The most likely reservoir species for MERS is bats, camels, civet cats, humans, or mosquitoes of the Aedes variety.
Unfortunate hairstyle choices.
F. Beluga whale. I have to remember that. Had that chance. But I won't edit one of them, and then you would know that that's the one that wasn't correct. Unless like, oh, don't, I forgot that was mine. Yeah, more correct, yeah. Pardon?
Yellow fever, we still don't really know what the reservoir is.
Actually, probably monkeys, non-human primates, almost definitely. At least for the sylvatic version, and otherwise humans, actually, because humans do a pretty good job of transferring. 10, 5, 39 people here today? Nothing like the nice weather, I guess. Okay, we're in the wrong window here. Do we like this? Yes, we like camels. The key here again is reservoir species. Just thinking about where the virus is probably circulating. I should say the viruses, because there are lots of them. It's generally circulating in the environment. What would the change that one would have to make to MERS to make A correct? SARS is almost definitely circulating in bats, and in fact, probably a whole bunch of other ones. Why civet cats? Because that's where we thought SARS came from originally, but it probably got civet cats from the bats. Humans are not a great reservoir species, at least not for SARS and MERS, because everything's pretty self-limiting in those cases. And Aedes are wonderful for all kinds of other viruses, but not these particular respiratory ones, because as far as I know, mosquitoes don't sneeze very much. So that would be this one. Other things as far as coronaviruses are concerned, the main thing here is, of course, what he puts in red, the replication, messenger RNA production, which is bizarre in what way? The nested subgenomic messenger RNAs. And so the fact that you have all of these particular messenger RNAs for the structural proteins, which of course you need a whole bunch of, and with those, then you have the polymerase, which is jumping back and forth. Whenever it hits a particular sequence on the negative strand, so it's copying the positive strand, it's a possibility to jump to the other end of the genome. And one thing that I forgot to mention last time is it's not just the cap structure, but there's also a 5' sequence, which is really important for translation, which is present at the 5' end of not just the genome, but also all of these subgenomic messenger RNAs. Otherwise, relatively straightforward as far as these viruses are concerned. So now we move on to the other side. Negative strand RNA viruses. So these are the mononegavirales, so the negative strand RNA viruses. Lots of interesting, nasty diseases caused by these viruses. Most people care about the filoviruses, which is Ebola and friends, who we'll be talking about next time. But turns out that measles, particularly worldwide, is way worse than Ebola by pretty much any stretch of the imagination. Mumps is also considerably worse, and then a number of these other diseases. Respiratory syncytial virus is a huge problem, particularly with neonates. Sendai virus and vesicular stomatitis virus are really good model systems that we use to study these things. In fact, I wrote a couple of grant proposals to study Sendai virus instead of measles because I didn't want to have measles in my lab. Rabies virus, of course, is quite well known for the nasty diseases which it causes, but also behavioral changes. And so a lot of people talk about how viruses cause behavioral changes. And so rabies is probably the best example of that. And then vesicular stomatitis virus is the relatively innocuous, if you're not a cow, way of studying these particular kinds of viruses. So Paramyxo and Rhabdo, they're basically identical to each other as far as we're concerned, i.e. at the molecular level, but they have very different virion shapes, which just goes to tell you that virion shapes basically mean practically nothing. But that's why they were named as such. So the Rhabdo are the rod-shaped, and the Paramyxo are the bizarre myxoviruses. The real myxoviruses are flu, which we'll talk about in a couple of lectures. A couple of important key concepts here. First is subgenomic RNAs, which are not made the nested subgenomic RNAs like for coronaviruses, but these are made in a very different fashion, and they kind of have to be because if you think about it, these are negative strand RNA viruses. So they're going to be made in a different way, and the way that that happens is what's called the start-stop procedure, which is where you make a short piece of copy of your negative strand, which is actually kind of nice because you copy the negative strand, it's immediately a messenger RNA. You don't have to go through an extra round. And if you have a particular mechanism for starting and stopping, particularly if it's only a certain relatively low frequency that you stop, you can start by making a whole bunch of one set of proteins and then less and less as you move down your genome. So we'll talk about that. Another interesting aspect of these particular viruses is that the RNA-dependent RNA polymerase has a really interesting stuttering mechanism and basically spits out a whole bunch of the same residues. And I give you one guess what those are. Where do you want to have lots of copies of one particular nucleotide? Say at the 3' end of a messenger RNA, you might want to have a whole bunch of adenines, exactly, poly A tail. So the other thing I wanted to mention here, we talked a little bit about this before, alternative start codons and, in fact, recoding for different kinds of proteins in a particular one sequence. FIX174 has this down, but it turns out that a lot of the, particularly Paramyxoviruses, do this quite a bit as well. And there's even some RNA editing, which takes place in some of these genomes. So as usual, our regular sort of outline here, origin, yeah, we'll talk about disease. And the mononegavirales, fortunately, almost no one calls them this because it's impossible to pronounce, unlike the picornaviruses. So, but again, it's just negative strand RNA viruses that have a single genomic piece. Structures are basically, all you need to know is here. That's what the Paramyxos look like. This is what the Rhabdos look like. Binding and entry is relatively straightforward, only we're getting binding and entry at the plasma membrane for the most part, except for these Rhabdoviruses here, which then go through our friendly endocytosis, pH change, membrane fusion process. Replication for these guys, and I probably should have put replication and messenger RNA formation in red here because that's the interesting stuff from the molecular point of view. Translation is pretty straightforward with these guys. They've got caps, they got tails. They're actually pretty easy to think about translating. And then getting out is, again, by fusion at the plasma membrane and then getting outside of the cell. So mononegavirus, again, pretty obvious. Mono 1, nega, negative strand, right? So pretty straightforward. Plasma membrane is where these are budding off of cells. And what this means is that usually the cells are actually not dying in this process. They're constantly producing new virus particles. One thing that I didn't put up the whole Baltimore scheme, what you have to have if you're a negative strand genome RNA virus, or at least negative strand inside the virion, you have to have a RNA-dependent polymerase, and you have to have a protein because you can't translate anything to make that protein. because negative strand can't be used by the translation machinery. So you have to package this RNA-dependent RNA polymerase. And they're always in the virions, and in many cases are actually even active in that particular virion. And again, on this start-stop messenger RNA production that we'll get back to in a little bit. Rhabdoviruses, these are famous for rabies, encephalitis, swelling your brain, really nasty. Fortunately, the progression of rabies is relatively slow, so you can actually vaccinate after you've been bitten by a rabid animal. And if you were lucky enough to be alive in Pasteur's days, the way that they would treat you is to take a bunny rabbit that had been infected by rabies and get a little bit of its spine and grind it up and inject you with it, which actually worked pretty well. It's better than dying of encephalitis. But fortunately, the newer vaccines are not made in quite the same way. But it was, in fact, a very effective vaccine, and one of the ways that people started thinking about using vaccines, and we'll get back to vaccine use in just a second. But this is what the virion looks like. Oh, by the way, just as a quick shout out here, this is the, what is it again? I think I've got a link on the next one here, but the Swiss virus site, which actually has these really nice cartoons of what these virions look like. So we've got a glycoprotein here on the outside, matrix protein between the envelope and your nucleocapsid, helical nucleocapsid that also has the polymerase protein bound to it at the end here, and then our N protein that's here as well. Again, rabies is the nasty disease. I wouldn't want to work with rabies in my lab either. So most people work with vesicular stomatitis virus, which is a virus which does cause nasty disease in cattle, but doesn't cause any disease in humans. So it's the lab, you have quote-unquote rat. One of the reasons people are so excited about using vesicular stomatitis virus, it turns out this G protein binds to very, very common extracellular molecules, and then gets picked up by endocytosis, and then causes membrane fusion to take place as those endosomes undergo the lowering of pH. And what this means is you can take this G protein and stick it onto basically any other virus and get good membrane fusion for taking place. And that's partly why people have in fact used this for probably the best Ebola vaccine out there, which is a recombinant, that's what this little R stands for up here. I'll get my pointer to work. Recombinant vesicular stomatitis virus, Zaire Ebola virus. And so it's a mixture of these two, mostly VSV, and then a couple of proteins from the Zaire Ebola virus. This was tested in the last couple of years, and the really interesting clinical trial that hopefully we'll have a chance to talk about next time does seem to actually protect quite well from disease, but fortunately there aren't any Ebola outbreaks happening right now, so it's kind of hard to test some of these vaccines. But that's the process, and I did also want to mention here briefly phase 1, 2, and 3 trials. Are people used to phase 1, 2, and 3 trials? Yes, no, maybe, shaking heads. So phase 1 is safety, around 100 people, so it doesn't make anybody sicker when you give it to them. Same thing is true with drugs. Phase 2 is to move up in numbers, usually hundreds of thousands of people, which is to optimize your safety and have a little bit of efficacy testing here. But the main thing is really show that you have protection from disease. And that's what happens in phase 3 trials, between 1,000 and 10,000 people, which if you've only got about 1,000 people who are sick with Ebola or hundreds of people, makes it really difficult to do these kinds of trials. But basically, that's where you get the efficacy, and at least as far as the FDA is concerned, it is true with most other regulatory agencies, is you have to go through phase 3 before you actually have any kind of approval, and you can actually start using these kinds of things. This process here, which I don't have, is sort of the $1 sign, $3 signs, $5 signs here, because they get much, much more expensive as you move through these processes. So we'll talk again a lot more about Ebola next time. Reason to come on Wednesday, or not come on Wednesday, as the case may be. So Paramyxoviruses, the best known of these is measles virus. Measles is apparently a very old virus. We have some indications, I'm going back and looking at mummies from Egyptian times where it looks as if they had measles legions. There's an extremely good vaccine, but despite that, there's still millions of infections every year. There's a sort of the cow version of measles, the rinderpest here, which is basically the second virus which we've managed to eradicate. So right after smallpox, and the eradication of rinderpest in the wild was, I think, about 3 or 4 years ago. So really impressive that this was able to be wiped out, particularly since the major issues were in the developing world, and so getting a good vaccine there was also really critical. Yeah?
Was the cow the host basically immune to the infection, and that's where, or were the cows, this is the cow version, right?
The rinderpest, yes.
So when they were vectors, did they show symptoms or were they asymptomatic?
Yeah, so this was a disease state, and so they did get very sick and died. And that process, excuse me, was something that was then treated mostly with vaccines. And so they made a decent vaccine for this and were able to use it to completely eliminate it, which is, again, really impressive in terms of getting rid of diseases. The measles vaccine, again, we'll talk a little bit more about it later on. The other major issue with measles is it's incredibly easy to transmit. So if one person in this room had measles, everyone else would be exposed to it. So it's really incredibly infectious. So that's probably why there are so many of these infections that happen per year and why vaccinate, vaccinate, vaccinate. So we'll continue beating on that particular horse for quite a while now. Mumps is another one of these paramyxoviruses. They're these emerging viruses. When I say emerging viruses, these are viruses that have reservoir hosts, other places, probably mostly bats, that also can cause some pretty nasty human diseases. You may or may not have heard of Nipah and Hendra viruses. The lab rat here, sort of like the VSV for, excuse me, Rhabdoviruses, is the Sendai virus here. And again, here's the link now, the viral zone. That was what I was trying to remember. Here for the Paramyxoviruses, they've got a negative strand genome in the middle here with a polymerase bound to it. And then instead of having this one G protein on the outside, the glycoprotein, these have hemagglutinin and fusion proteins, but still have a matrix protein and an N protein bound to the genome. So let's take a quick aside here and talk about why everyone should immunize. Why? Why should you get the measles vaccine? It's incredibly effective. And the reason for this is between 0.1%, 0.2%, so 1 in 1,000 to 1 in 500, kids who get measles actually die. It's a pretty high number. How many of you have read Roald Dahl? James the Giant Peach, etc. Charlie the Chocolate Factory. His daughter died of measles from nasty encephalitis, which happened there. 1 in 20 will get pneumonia, and this is probably why most of there's death which happens. Now there are side effects. 4 in a million kids get vaccinated, have some serious side effects. Almost all of these are allergies. And so that's like 0.0004%. So relative to this, about 2,000 times less likely than death, you'll have an allergic reaction. I think the numbers are pretty clear there. But actually thinking about humans are really bad at risk assessment. Really, really, really bad at risk assessment. It's probably evolutionary because we had to deal with these small communities that we were with. But I think 2,000 times more likely to die than get an allergy, I'm going to vaccinate my kids. Before there were vaccines, there were, in the US anyway, 4 million cases a year in the US of measles. Almost 200 cases in 2013. 90 to 98% of these cases were unvaccinated. So again, thinking about these things, measles was eliminated in the US in 2000, but still we've got a number of these other cases, 2013 here. Let's take a quick look at this. This is a really nice, in fact, graphic, not quite novel, but novella maybe, on vaccines work. Diseases were practically fairy tales before the anti-vax movement. Whooping cough and measles. 2008, 2 outbreaks of whooping cough. 2014, measles, multiple outbreaks of whooping cough. Vaccine coverage. This is according to the CDC as of last night. It's pretty scary that Texas, and particularly Mississippi, the darker colors here are way better at vaccinating than Washington, Idaho, Oregon, and you'll also note here California. So Texas and Mississippi are 99% plus vaccination for kids. We're at about 94, and California's about 92. And why is this so critical? Many of you may have heard about the nastiest place to go to get measles. That would be here. Oh, sorry, back up. Magic Kingdom, of course. That's where you go to get measles, right? In 2014, 2015, there was a big outbreak, about 200 people or so. Ended up getting sick probably because of one case of measles that was brought here from the Philippines. There was a big outbreak in the Philippines in 2014. That then was brought presumably Disneyland and then spread. Almost again, all of these cases non-vaccinated. In the world, because unfortunately vaccines haven't done terribly well there, 150,000 people a year die from measles, and again, about 20 million or so are infected. So measles is a big deal in the rest of the world, and we are probably rather complacent as far as that's concerned. Yeah, sure.
How closely related are the German measles, like the rubella, to the rubella? Because I was vaccinated against the rubella, and I still got the German measles, and everyone in my town got it.
Yeah, so there's measles, mumps, rubella. So rubella is unfortunately the worst of those vaccines in terms of the protection. And so that's a potential issue. The measles vaccine is actually the best of those. So it may well be that for some reason, maybe because it wasn't stored properly, you certainly will have outbreaks, and some people do in fact get. And if you look at one of the things, so MMR, how many times do you have to take it? How many of you have kids? You know how many times you need to take it? Twice. First time is about 90% effective. Second time is about 97% effective. So, and there's still 3% where it doesn't work.
I heard the virus, though, I don't know, this website that may not be, this paper that I was looking at, says the viruses aren't very related, the rubella versus the rubeola.
Okay, yeah, so okay, let's maybe back up a little. German measles and measles are completely different viruses.
Okay, that's...
They have no similarity whatsoever. They just happen to be in the same vaccine. So, but yeah, they're completely different in terms of the actual virus. So if you've been immunized against one, then it doesn't provide any protection against the other, and vice versa. Most of the time, they're formulated together. So, and there may be some cases where that hasn't in fact happened, and there could be some issues with that. Yeah.
How often does the vaccine itself, get changed? Does the virus have like a low evolutionary rate, or is it like the flu vaccine?
Because the question is basically how often do you need to change the vaccines in this case? The Edmundston strain, which is used for measles, I think was developed in the late 1950s, is still being used and still works great. Quite why that is, and that apparently whatever is necessary for getting the measles to function properly, and that antigen effect is good enough for that Edmundston that it hasn't been able to escape whatever the antibodies that are generated from it. But it's a great question, because there are clearly a lot of changes that happen. One of the big differences when we talk about flu, again on Friday, another reason to be here on Friday, there's a lot of other things that are going on with flu that are above and beyond what happens in terms of these mononegavirales. So that's probably why. But that's also to some extent a little bit of a guess. Okay, so let's take another quick look at what's going on. why the US, you still need to vaccinate here. That's because people come from other places, particularly places like India and China, which have very large numbers of reported cases here. Also places like Nigeria and Ethiopia, a number of places here which aren't even reporting. Philippines, also very large numbers, and this is this last season, September 2015 to February of 2016. A lot of the imported cases in the US, you can actually look at the genomes of these particular viruses because they do change quite a bit. Most of the imports are from nasty places like England, France, and Germany. In fact, there was a big mumps outbreak to someone who was in Switzerland and caught mumps there and brought it back. So that's not a, there's no safe place to go. Yeah, other than maybe Greenland.
And most of the Nordic countries
Yeah.
are listed as no data reported to. They not have state vaccination programs.
So I suspect here that it's they may or may not have these. I don't know. These are just data I pulled up last night, 5th of April from 2016. And also a lot of countries in Eastern Europe haven't been reporting these data, and so I'm not quite sure why that is. Usually measles is reportable, and certainly is in the U.S. And it may be that they just hadn't, sent their data to the WHO as of February of this year. Maybe they send them quarterly or something like that. That I don't know. This is what happened basically with that outbreak in Disney World. Oh, no, sorry, Disneyland. No, it's not. That would be over here. So Disneyland here, and you remember also that state where the vaccination levels were low. Most of the cases were in California, some of the neighboring states, and then a few of these across here. And this is all then from that one particular outbreak that happened. And I love this, an amusement park in California. I guess they're being politically correct down at the bottom here. All of 2015, actually up through April 24th of 2015, there were also some outbreaks in Illinois and Florida. Any idea why there were these pretty big number of outbreaks in Illinois? So a lot of these are in the Amish population, people who also don't vaccinate. And there's been a really great push that's happened, in fact, in a lot of these communities to try and get the locals, actually members of that community, to really go out and try and get a lot more vaccination. And that may well be why this year we've only seen 6 cases, which is really pretty amazing considering that this time last year were 166. So hopefully these bars are going to go even further down. One interesting aspect here of this 2015 bar, which is relatively low, you notice, relative to 2014. Some of the 2014 numbers are from the Disneyland outbreak, but a lot of these are also from an Amish outbreak. There are about 300 people, unfortunately, who came down with measles. Now, one of the things that you'll also notice here is the numbers, about 600, a couple hundred here. How often do you have people die from measles? About 1 in 1,000, 1 in 500. So in fact, fortunately, no one has died in the last few years of measles, but that may well change, although this is an extremely good sign here. I was blown away that there were only 6 cases that have been reported so far in 2016, again, relative to last year. Okay, questions on this, and if he says that, what does he mean? It means we're about to have our own questions here. So, most cases of measles in the USA, I should say in the last couple of years, were introduced from outside the US when people who are not vaccinated killed the people who were infected, caused encephalitis A and B.
The lecture doesn't seem to be on D2L.
I may well not have posted it, that's correct. I think I made the copies, but I didn't post it yet. I'll do that. It's Monday. It was a nice sunny weekend. They're there. I've got the PDF and the PowerPoint. They're sitting on my computer. I just couldn't get them uploaded. This is why you need to listen, Peter. Up to 45. A few more people came. 10. Okay, so I'm willing to bet that the person who selected B actually has a really good reason for it. They weren't trying to mess with me. Because when I realized reading this question, you could actually understand it as being the cases that actually happened in the US are probably from someone who was in the US and then infected people. So I will go and find out who that person is and give them credit as well. So it's one of the nice things I can do with the software here, just to check that. Because I was looking for A and B, but I do see how you could interpret the question as being B as well. Because, yeah, that's, i.e. not having A because, the individual cases that came into the U.S. were probably, each of the outbreaks, for instance, that we had last year, there were 5 outbreaks, there were probably 5 people who came into the U.S. With that, whereas there were hundreds of people, or almost hundreds of people who were infected. So I will take B as well as an answer for this. But I was looking for E. So I know I'm not going to insist that it was B for everyone else. Okay, so now finally we can talk about the interesting stuff, i.e. the molecular biology of these guys. Paramyxoviruses, they show the nice cartoon, nice spherical, virion here. They're usually not. They're usually much more like blobs. And that's true for almost all of the enveloped viruses. They're really pretty blobby. One of the things here that makes a big difference from here in the coronaviruses is there's no really obvious spike sticking out here on the outside. It's a pretty uniform structure on the outside. And we'll see this is also very true for the orthomyxoviruses, the flu, a little bit later on. Turns out that the virions look practically identical to each other. Big difference is when you take a little bit of detergent and break open the membranes here, you have these nucleocapsids, which are RNA that's complex with the nucleocapsid protein. And so these really long filaments, maybe a little bit hard to see here, but this is a helical structure with the proteins on the outside. And if you look here, here's one looking end on. You can see there's a hole in the middle with the virus proteins around the outside. A few of these paramyxoviruses will present, i.e. virions, that are much more filamentous and elongated. And again, this is a lot like what you see with the flu viruses, the orthomyxo, the real myxoviruses. Rhabdoviruses, here's a nice example of a rhabdovirus, bullet-shaped or rod-shaped. The rhabdo is just from the rod-shaped. Here you can see the projections. This is the G protein around the outside, where this is what's going to bind to the sialic acid on the outside of the cell and cause endocytosis. You just release the genome from here, and now we've actually gotten rid of most of those viral proteins. You can see this nice genomic RNA that's present here. One of the things that you see with the rhabdoviruses is that the nucleocapsid, so i.e. the protein bound to nucleic acid, is also directly associated with the M protein, and that matrix protein, again, immediately under the membrane, and that's probably why you have these, I like to think it's more of the rod-shaped structures rather than the more spherical or non-rod-shaped, probably is a better way of talking about it, form that you get for the paramyxoviruses. Again, this is your standard paramyxovirus. In the middle you have a single genome. It shouldn't be detached here in all these places. All this stuff should be connected, which has the N protein, the polymerase protein, and a phosphoprotein. All three of these proteins are present here in the virion. The matrix protein right here, again, is sort of providing a link between the envelope proteins, which are sticking through the membrane, and the nucleocapsid protein here on the inside. And then for the paramyxoviruses, you have the hemagglutinin and fusion proteins. Some cases, there's a neuraminidase. We'll talk a lot more about this when we talk about flu on Friday. But hemagglutinin will cause blood cells to bind together, so the hemagglutination assay is also a good way of detecting a virus infection with measles. In fact, a lot of cases, that's what's also being used for. So let's focus in on these fusion proteins in a little bit more detail. This is the fusion protein itself. As we talked about way back when we talked about membrane fusion taking place, and we'll again talk more about this when we talk about influenza as well. These fusion proteins almost always have a fusion peptide in them, which is extremely hydrophobic. That means it loves to stick into membranes. But in order to make sure that this fusion peptide is not stuck into a membrane while you're making the virion in the first place, it has to be protected somehow. And usually that protection is in the form of some kind of proteolysis, in this case also a disulfide bond formation, which basically hides that fusion peptide from whatever membrane it's associated with until you have interaction, usually with this hemagglutinin protein, with whatever it's going to be binding to on the outside here. For the paramyxoviruses, this is binding at the plasma membrane. For vesicular stomatitis virus and probably rabies as well, this fusion peptide is part of the hemagglutination protein. So this is called the G protein. G just stands for glycoprotein. So glycoprotein, heavily modified with sugar, is going to be the outside of your virion. It has both of these activities, both the fusion activity and the hemagglutination activity. And again, VSV goes through endocytosis as opposed to what happens with the paramyxoviruses, including measles, which all then fuse the plasma membrane. Because this VSV-G protein, the glycoprotein from VSV, is so good at getting those virions into endosomes, and most cells undergo receptor-mediated endocytosis. What a lot of people have done, and even in some development of, in fact, some slightly different Ebola vaccines, is take this VSV-G protein and put it into a different virus, and a virus that would otherwise be binding to different cells, to different receptors on the outside of the cell, and then just put this G protein from VSV in there instead. And what that means is whatever that recombinant virion is will then get incorporated into the cell. Fusion will take place, all again because this G protein does both binding and fusion. And that way will release whatever genome you happen to have on the inside here. And that's what's called pseudotyping. So usually that you type a particular virus by figuring out what cells the virion binds to. You throw on VSV-G protein, it basically can bind to more or less anything. Yeah?
Wait, so does that attenuate the virus?
So the idea, so the question is, does that attenuate the virus? So in this case, usually attenuation is you take a wild-type virus and then infect it through multiple rounds on a particular host. And then you will eventually accumulate mutations, which leads to lower virulence. So I wasn't going to talk about that, but that's the whole process of attenuation. That's what Pasteur did, and in fact, it wasn't the first rabbit that you got the spinal cord from. It was actually after a whole bunch of rabbits that had been infected in that process. So he, in fact, is probably the one who's, well, I see the one who's credited with coming up with that attenuation process. But that's a wild-type virus. Here, this is recombinant. This is taking in this case, the glycoprotein from VSV and putting it into a completely, totally, and utterly different virus. And classically, this is actually used, I mentioned gene therapy right at the beginning here, for retroviruses, because retroviruses are really good at inserting genes into genomes. But retroviruses usually have very narrow host ranges. There are only a few kinds of cells that they can go into, whereas these VSVs can go into pretty much any cell because they have a really common receptor. Those can then be bound and be brought into the cell. Okay, so that's the whole idea of pseudotyping here, and most of the time when you're talking pseudotyping, you're talking VSVG, although pseudotyping is a much more general idea. You can take the receptor binding protein from any virus and put it into a different one, again, through recombinant techniques, and that would be a pseudotyping arrangement. And we may or may not have a chance to talk about that when we talk about retroviruses later on. The other protein that I wanted to talk about specifically here is the Nucleocapsid protein. This is what binds to the genome, and it binds in a very specific way and gives you this sort of super helical structure here. And what's important about this is, unlike what happens, remember TMV, this the RNA was bound sort of on the inside of the particle. Well, tobacco mosaic virus is a naked particle, so it's actually protecting the RNA. Here, the RNA doesn't need to be protected because it's inside an envelope. And so here, the packaging leaves the genome on the outside. And one of the things that people found when they studied these is that it's literally every 6 nucleotides that you have one of these nucleocapsid protein bound, and then it wraps around in almost exactly 80 nucleotides each time around. And here is the 3' OH, and you remember this is the negative strand genome, and so this 3' OH here is where you have the first set of nucleotides. Count your way along here. And so these nucleotides here, as it turns out, are also really good promoters and important for positive strand replication, so the messenger RNAs that you get, but also nucleotides which are right here, about 80 nucleotides away, turn out to be critically important. And then these are those, again, the actual sequence here is not critical, but you do have nucleotides. They're about 80 nucleotides apart. What that means is it looks as if the RNA-dependent RNA polymerase actually will bind to not only these sequences here, but also these sequences here. And that means is it can only use genomes which are packaged in this form as templates. And this is a really good way for the virus to know, again, totally over-anthropomorphizing here, that this is a genomic RNA as opposed to a messenger RNA. And so it's a way that the viral polymerase can differentiate between messenger RNA and genomic RNA. And so that's the important aspect of this particular protein. So that's the glycoproteins, the H proteins, the N proteins. Then we'll talk much more about the polymerase a little bit later on. But before then, we'll have one more clicker question, which is, which of the following rhabdovirus proteins is most likely to be used to make an Ebola vaccine? N, C, V, L, or G? Clicker question in a different place in the lecture. Oh well.
Or a little bit later.
Yeah, later actually. Yeah. Oh well. 10. This time I think somebody's trying to mess with me. So yeah, the answer I was looking for is G, the glycoprotein, because you can have pseudotyping. Why would pseudotyping be useful in terms of making vaccines?
More general.
Get to bind stuff that normally wouldn't, but one of the things that people need to use for vaccines, you need to make a lot of this virus. And so if you're making a whole bunch of it, it's really nice to be able to get it into lots of different cell types. So that's one of the reasons that people particularly like to use these as vaccines. Okay, so sort of mentioned fusion already. Sendai virus, which is the model system. Yeah.
I just want to make sure, so by making a lot of it, you mean that it increases the efficacy of the vaccine? Is that what you meant by that?
No. So when I say making a lot of that, what I mean, thank you for asking, is that in the production of the vaccine, you need to make lots and lots of virus to then be able to inject into someone. And for that purpose, you want to have lots of potential different cell types that you can use to make that process. Yeah.
So, like, typically the retroviruses are being very picky about the surface proteins they want to deal with.
So yeah, so the question here is maybe we haven't talked too much about host range or typing. Retroviruses, but if you think about the coronaviruses as well, are also very specific about what they go into. And here, measles is also a really good example for that. Measles uses immune cell receptors. And we talked about this already. I should say Alec talked about this a little bit already when you have to think about the Zika, dengue, etc. These are also binding to a lot of immune system receptors on the outside of these cells. And so that process often is very, very specific for the different viruses. Varies, but that's one of the main ones. Yeah?
Does that mean that people with better immune systems often get worse infections?
Okay. So question here, do people with better immune systems get worse infections? We'll talk a little bit about that when we talk about flu. There's sometimes it's the reaction to the virus as opposed to the virus itself, which causes most of the problems. And so, but there are also issues if you have a weaker immune system, then it's going to be a problem. So that's anytime I try and give an answer like this, I'm treading on really, 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 really thin ice. And I think the answer is yes and no and both. for questions like that. But yes, it's a very interesting question, and certainly in the case of the 1918 flu, there does seem to be a correlation there. But yeah, we'll hopefully talk about that on Friday. Friday's lecture is only going to take about 4.5 hours. That's all right, isn't it? So yeah, how do we have binding? Sendai virus, and again, almost definitely rabies virus as well. Was it bind to sialic acid? Sialic acid is a disaccharide present on the outside of practically all cells, so it's also a really good way of binding and looking at these particular viruses. On the other hand, measles is very specific to immune cell receptors. To do this, you need first hemagglutination, and then F. What happens to F? If you're a Paramyxovirus, it's just when you bind to a receptor. If you're a Rhabdovirus, it's again when the change of pH happens. These are almost all trimers, as we've seen before and see again. Quite why trimers is an interesting question. I don't think anybody really knows. But that seems to be very common in terms of these type 1 fusion proteins that happen. Once you have fusion, you release the genome, and that genome looks like this. And this is partly why I say that Paramyxos and Rhabdos are basically identical to each other, because their genomes are practically identical to each other. The only real difference, again, is in terms of the shape of the actual virion. Again, it's a negative strand, single molecule RNA, and has everything that you possibly would want. So basically all of the proteins that are encoded in the genome are present in the virion as well. So N protein here, nucleocapsid protein. P, C, and V. P is a phosphoprotein that's associated here. Matrix protein, fusion protein, hemagglutinin neuraminidase protein, L protein. The L protein is the polymerase, and that's true. You'll see this for all of the Paramyxo and Rhabdoviruses. The L protein is the polymerase. And here, just as a reminder, F and HN are in G in these rhabdoviruses. So if you look at the genome, again, it goes from 3' to 5' because we're a negative strand virus. The interesting thing here are these intergenic segments. So the intergenic segments have a termination and really short sequence here, and then a start, which are about 30 nucleotides apart. And one of the things that you'll notice here is that there are 5 uracils here, and again, this is the negative strand. What does that mean? When you copy it, it's going to be 5 A's, right? Yeah, good. So that happens at the end of each of these genes: the N gene, PCV gene, the M gene, the F gene, etc. Think about it. Nucleocapsid protein is the one that you need the most of. PCV you need a little less of. Matrix protein you need a little less of. You need a little less of your fusion hemagglutinin proteins and the least of your polymerase. So you need the most of this and going down in terms of the amounts that you in fact need. How do you get this? It's all about the L protein and how the L protein works. L protein, again, is the RNA-dependent RNA polymerase. Together with the P protein, it will bind to the 3' end of the genome, and again requires sequences which are a little further away because of that wrapping structure. And then will transcribe and gets to this intergenic sequence. The first thing that happens is that this polymerase makes lots of copies of those U residues. This is a stuttering process. So it'll make 5 A's, and then make 5 more A's, and make 5 more A's, and make 5 more A's, and make 5 more A's. And in some cases, the polymerase will then fall off. Other cases, it will keep going and restart right here and make a copy of the next gene, PCV in this case, stutter a little bit, sometimes fall off, sometimes keep going. Then it will get to the next gene, the M, etc., etc., etc. And this X here is just basically to show that if you make a mutation in this gene which causes the polymerase to fall off, you never get the rest of the genes over here. And why was that important? Oops. The reason that that was important is because this showed that the single entry model, i.e. that the polymerase came and bound once and stayed on the template, was the correct one. One of the other models would be that, okay, the polymerase comes and binds to the start of each of these transcripts separately, comes out from the outside. If you have a mutation here at the end of this gene, you would still be making these proteins. But if you make a mutation here, you actually don't make any of these proteins here. And what that tells you is the polymerase actually stays on to the template and makes all of these positive strands, which are these black bars here, in terms of making the proteins. So this is the process again. Starts at the 3' end of the genome and will go through, make lots of copies of each of these individual messenger RNAs. How then do you switch from transcription to replication? The key to switching here is the concentration of the N protein. And remember, the N protein is the very 3' end of the genome. That's the one that you're going to make the most messenger RNA from. And so once you have enough of that N protein, it binds to the genome. And as more and more of that accumulates, they're going to end up with more and more of your genome, which is present with the N protein. And so what will happen is you'll switch then. The polymerase will switch from making messenger RNAs to making genomes. And so that process is basically shown here as a cartoon. When you start out, this is what you start out with right here in the middle. Here's your L protein that will make messenger RNAs from your genomic RNA. And start and stop and start and stop and start and stop. Until you get enough of this N protein. As soon as you have enough N protein, you switch over and then start making the anti-genomic RNA, which is now a positive strand for this whole genome. And then as long as there's enough N protein, this will now serve as a template to make more negative strand. So it's all about the concentration of the N protein. If you have RNA which is not bound by N protein, you're going to keep making your messenger RNAs. As soon as you accumulate enough of the N protein, then you switch over to replicating the genome. So it's all about N and N binding to your genomes in terms of making genomes. One other thing that I wanted to mention here is PCV. You've always wondering why is there one gene that's called PC and V. Basically, this is just because this particular messenger RNA can be translated in multiple different reading frames, and you have some stuttering that happens in the reading frame. So different reading frames here, the C protein, the P protein are in different reading frames, overlapping open reading frames. It should sound really familiar from 5X174. We've got overlapping open reading frames. The other difference here with P, C, and V is that the polymerase, the L protein, not only can stutter when it hits U residues, it can also stutter when you get C residues in the template. And what happens there is you change the coding sequence here at the C terminus of your eventual protein. And so just adding some extra residues here, also known as RNA editing, that way you end up with different proteins. So in this case, you've got, well, actually multiple different proteins here, but it's always called P, C, and V. But the main protein which is made, which is your standard start codon, no stuttering. The C protein, which is made from a different start codon at this end, and the V protein, which is made due to stuttering at the end of your, say, the coding sequence, which is present here. So a lot more P than C and V. Turns out there are other proteins that are made here. It's not clear if they're ever useful. The W protein, C prime, Y1, and Y2, they are made, but it doesn't seem to be, they don't seem to be important for actual virus function. I mentioned this already. Basically, it's the same thing. N binds the genome, and you don't get any stuttering anymore. How do you put these virions together? Again, they bud at the plasma membrane. So they come in at the plasma membrane. They're made at the plasma membrane. You have your M protein together with your nucleocapsid protein that interacts with your matrix protein. Matrix protein, again, is the sort of bridge between nucleocapsid and all of your membrane proteins. That will bud off, and now you have a virion which can go off and infect other cells. One thing that I also wanted to mention here is that very often what happens here, because of the presence of these fusion proteins or G proteins, is you'll end up with a syncytium formation. What's a syncytium? Multinucleated cell. And why is that? It's because all the membranes have fused. And the reason the membranes have fused is because you have all of these fusion proteins sticking out on the membranes of one cell. And so since they're forming at the plasma membrane, then the plasma membrane here, instead of budding off to form a virus, could also fuse to the cell that's right next to it. And in fact, people use the G protein, VSV G protein, in many cases to actually literally form syncytia. So you can take that G protein, put it into a cell, and those cells will then fuse and make multiple nucleated cells, or basically one continuous cytoplasm, just by expression of this G protein. And then just as a reminder here, the genomes of all of these mononegaviruses have to be a multiple of 6 nucleotides. Why is that? Because the N protein binds to 6 nucleotides. And it turns out if there's any change, +1, -1, anytime you have a non-bound piece of RNA, that genome is going to get degraded. It's not going to be functional. And so any of the changes, that may be why you don't see so many changes in these paramyxoviruses as you do in some of the other viruses like influenza, for instance. The rule there is not as strict in terms of absolute numbers of nucleotides. So that may be part of the reason. And again, that's a hand-waving argument here. But that's the process. So those are our measles and rabies viruses, or paramyxos and rhabdoviruses. Again, we talked a little bit about disease and vaccinate, vaccinate, vaccinate. How many times are you going to say that this term? The structures are pretty straightforward. Binding and entry, again, just a big difference between these two. The rhabdoviruses can bind and enter pretty much anything through mediated endocytosis. These paramyxos are much more specific. Again, most important here is probably that start-stop mechanism for messenger RNA production. And then it's the concentration of the N protein that leads to what genome replication you have versus messenger RNA production. So with that, next time we'll talk Ebola, other nasty mononegaviruses, which actually kill a lot fewer people than measles, but that's a different story, and we'll talk about that on Wednesday.